Hi everybody, welcome. Thank you for coming to my talk. I'm Bertrand Black and I'm excited to be here today to tell you how target capture sequencing revealed hidden diversity in the lady fern or Athyrium filix-femina species complex here in the Americas. I'm a PhD candidate in the Barrington Sundu Lab at the University of Vermont, where we study fern systematics, biogeography, evolution, ecology, as well as biodiversity. For my research, I chose to study the still unresolved Athyrium filix-femina species complex. These ferns are distributed across the northern hemisphere and down into South America, where they are characterized by a terrestrial habit with highly dissected fronds, swollen petiole bases with dark brown, black, and sometimes even gold scales, and sporangia that form diagnostic J-shaped clusters on the underside of their fronds. Originally described by Linnaeus, Athyrium filix-femina was thought to be a single species with Eurasian origins and a widespread transcontinental distribution. However, while exploring the Americas, European botanists came across lady ferns which seemed familiar, but they didn't really recognize. And over many years, American taxa were described as either new species or as intraspecific variants of the familiar Eurasian plants. So with all this diversity already described on the continents, how did we end up in our current situation where most modern floras only recognize a single widespread Athyrium filix-femina? Long story short, not everybody agreed, especially with regard to the number of Athyrium taxa in North America. Prominent botanists of the 19th and 20th centuries thought that the Athyrium in the Americas was too morphologically similar to its European relatives and did not warrant recognition as distinct species. However, they were working with a limited sample size and had not yet encountered the full range of its diversity on the continents. There was also no consensus on the number of American taxa, the taxonomic rank they should be treated, or the efficiency of the characters that can be used to distinguish them from one another. This debate culminated with the publication of the Flora of North America, where in it, all American Athyrium are treated as a single species with 4 endemic North American varieties. In 2018, Ranway placed the lady fern genus Athyrium in phylogenetic context for the first time using plastid sequence data. They found that it contained 10 large clades, which he organized into 10 named sections shown here. In this study, they had a global sampling strategy, but only included 3 accessions of Athyrium filix-femina from across its huge range, 1 from North America and 2 from Europe, which are shown here in green and blue, which were recovered in the type section Athyrium. However, even just these 3 specimens were important, as the tree recovered the species Athyrium filix-femina as non-monophyletic by the inclusion of the other Asian taxa shown here in red. While plastid sequence data has been historically used in plant phylogenetic studies for the last 20 plus years, this type of sequence data has its limitations. This led to the integration of target capture sequencing approach into plant phylogenetics, which takes advantage of next-generation sequencing technology and utilizes a custom lineage-specific probe set to seek out and amplify particular regions of the nuclear genome for phylogenetic comparison. Until recently, this technique was limited to flowering plant studies, with the most widely used toolkit probably being the Angiosperm 353 probe set. Fortunately for me, in 2021, a new universal probe set was released by the GoFlag project for all flagellate plants, such as ferns, lycophytes, and gymnosperms. That very same year, another member of the GoFlag project and fellow graduate student in my lab, Susan Fawcett, showed in her research that this approach worked amazingly well in sorting out the still unresolved genera of the Thyllipteridaceae or marsh fern family. So with this in mind, for my research, I wanted to evaluate this approach not at the generic level, but in a much more closely related group of taxa, such as members of a species complex. For this work, my goals were to first generate a representative sample of Athyrium filix-femina from across its wide range, evaluate closely related species lineages using the target capture sequencing approach and next-generation sequence data, as well as investigate hybridization, produce a revised taxonomic treatment and resources for Athyrium species in the Americas, and ultimately propose a biogeographic hypothesis for the origins, migration, and dispersal of Athyrium to the continents. I'm happy to report that after a few years of sequencing, we are finally happy with our coverage of the currently recognized Athyrium filix-femina range, seen here in the lower right-hand corner. The points in red represent our current sampling and study system. In addition to covering almost the entire geographic extent of this complex, this system includes a comprehensive assembly of morphology, habitat, and nomenclature. To construct our phylogenies, we extracted DNA from herbarium specimens for sequencing and built our trees using two different approaches. The more traditional maximum likelihood approach using a concatenated supermatrix of the aligned sequences, which we performed in IQ-tree, and another more modern approach using a multiple species coalescent model, which not only takes into consideration SNP data, but gene tree topology as well, which we constructed in Astral. Both models have their own assumptions, limitations, and advantages, which unfortunately I don't have time to get into at the moment. But by applying both approaches, we are able to compare methodologies and gain the most complete picture of the possible evolutionary relationship between these taxa. Using the recovered clades as a roadmap, we could then use georeference collections data from the preserved herbarium specimens in conjunction with bioclimatic and soils data to get a sense of their environments and the geographic range on the landscape. We then applied a similar approach to using our recovered clades as a roadmap to explore the morphological space across the groups and determine which, if any, morphological characters could be used to diagnose whole species lineages. These include discrete and quantitative metrics, as well as scanning electron photography of microscopic features such as spore ornamentation. So let's see what we found. Looking at the concatenated maximum likelihood tree, we recovered monophyletic genera within the Athyriaceae family with strong support, and confirmed the relationships previously established using plastid sequence data. We also found evidence for the 10 named sections in the genus Athyrium previously shown by Wei, obviously with a much larger sampling of the American taxa across their distribution. We recovered taxa from temperate North America and Eurasia in section Athyrium, as previously demonstrated with plastid sequence data, but interestingly found that neotropical Athyrium were in a completely different clade, section Mackinoniana, which previously only contained Athyrium from tropical Asia. If we take a deeper dive into this biogeography and color each of the major clades we recovered, we can see that there is a clear geographic structure with discrete ranges from each of the taxa in North America and Eurasia, which we did not find in the neotropical taxa despite sampling from a wide range of morphology, habitat, and nomenclature. Now, I have not yet completed my formal biogeographic analysis, but the emerging picture is interesting to say the least, as this topology suggests that there are two independent dispersal events of Athyrium to the Americas from their ancestors in Asia, one to the neotropics and another to temperate North America. In addition, and what I think is the most exciting implication of this result, is that Athyrium taxa from Europe and Asia are nested in an entirely American clade, and suggest that their ancestors were of American origin and not Asian origin as previously thought. This would mean that from a common ancestor in the Americas, these taxa diversified across Eurasia and subsequently America as well on two separate evolutionary tracks to eventually form the distribution of the American and Eurasian taxa that we see today, though the timing and directionality of this migration to the Americas remains to be determined. We also found that not only are these taxa distinct both phylogenetically and geographically, but they have unique environments and habitat preferences in which they occupy on the landscape. The results from our ecological models can be seen here in the form of two principal component analyses. In the figure on the left, looking at bioclimatic data, we can clearly see non-overlapping pattern, where each of our recovered lineages occupies a different place in the environment with few exceptions. These differences were due primarily to changes in annual temperature, precipitation, and seasonality. In the figure on the right, we see the opposite pattern, where almost all of the clusters are overlapping. This indicates to me that soil characteristics are generally shared across all taxa. When comparing the two different tree building approaches, we recovered the same major groups, but found that there was a topological shift at the deeper nodes in one of the major lineages found from the American Southeast, Athyrium splenoides. In the concatenated tree, we recovered it as sister to the northeastern taxa, but in the multiple species coalescent tree, we recovered it as sister to a clade consisting of both the northeastern and western taxa. However, our multiple species coalescent tree was not recovered with a high degree of posterior probability at these nodes, and to me this indicates that there is a high degree of gene conflict between these four lineages. This could be due to a variety of factors such as introgression, incomplete lineage sorting, or a lack of phylogenetic signal from these loci. At the moment, we are still compiling and processing the vast amount of morphometric data that we've assembled from each of our accessions, but I'm happy to report that the emerging picture is that the morphology matches the phylogeny as well. With the addition of this morphologic data set and the others that we have discussed here today, our research has found that Athyrium is more diverse in the Americas than ever thought possible. We have not yet settled on the specifics of the nomenclature for each taxa, but are looking at recognizing at least these colored clades at the species level, each with diagnosable morphology, habitat preferences, and distinct geographic lineages. One final unanswered piece of this story is to assess the amount of gene flow between taxa and look for evidence of hybridization and polyploidy. In a preliminary investigation into this, using our target capture sequence data, we used the metrics of locus heterozygosity and allele divergence to look under the hood and identify this individual from Mexico as a punitive, hybrid, or polyploid taxon. As you can see in the top right of the figure on the right, this individual was found to have a unique genetic profile compared to the other taxa in the complex. And this initial evidence is corroborated by its large spore size, which are often found in polyploid ferns. Now, a logical next step would be to see where this individual comes out in our phylogeny. And interestingly enough, it is recovered on a lone branch, sandwiched between the more temperate section Athyrium and tropical section Mackinoniana. It is even found where these two distantly related lineages converge in the transverse mountain ranges of central Mexico. And it is my hypothesis that this plant is the result of a hybridization event between these temperate and tropical lineages of American Athyrium. In order to show this conclusively and identify its progenitors, the final step will be to perform allele phasing, to which target capture sequence data is uniquely adapted, and confirm or deny this hypothesis. So the big picture is that I'm hoping that this research will serve as a model to accelerate the documentation of still unrecognized fern diversity in the Americas and elsewhere using contemporary and integrative approaches to phylogenetics. The study will also generate and share revised taxonomic resources for American Athyrium taxa that can be used by land managers and conservationists to better protect biodiversity in a changing climate. And finally, this kind of research provides a unique opportunity to advance our current understandings of the origins and evolution of floristic diversity in the Americas. And with that, I'd like to thank my amazing advisors, David Barrington and Michael Sundu, all of the amazing members of our lab, both present and previous, my committee, who I could not have done all this without, as well as Carol Kaloff and Alejandro Vasco, who have helped me tremendously with this project, and the team of undergraduate researchers who were just excellent and helped me generate all the data which I shared with you today, as well as the contributing herbaria where I sourced all of my collections, and the funding sources. Thank you so much for your attention and time. Happy Evolution 2023.